Welcome to Science in Focus, a free public lecture series showcasing the latest research from prominent UTS scientists. I'm Jim McNamara, Deputy Dean of the UTS Faculty of Science. In accordance with custom, I would like to acknowledge the Gadigal people of the Eora Nation, upon whose ancestral lands our city campus now stands. We pay our respects to their elders, past and present, and acknowledge them as custodians of traditional knowledge about this place. Today it is my pleasure to introduce Dr. Lana McClements, who is speaking on the miracle of pregnancy and new life. Lana is a lecturer in biotechnology in the UTS Faculty of Science. She researches women's health with a particular focus on understanding pregnancy complications such as preeclampsia. Her research has had significant impact in creating new monitoring strategies for women who are at risk. We are pleased to present Dr. Lana McClements.
Hello, I'm Laura McCaughey and I'll be the moderator for today's event, collating questions and asking our speaker, Dr. Lana McClements, questions both during and at the end of her talk. I'll now hand over to Dr. Lana McClements to hear about her fascinating research into pregnancy and its possible complications.
Thank you very much, Laura. Hello, everyone. I'm delighted to be able to talk to you about pregnancy and also my research, which is in relation to pregnancy complications. So I'd like to start my talk by showing you a picture of myself with my second-born daughter. And this is 2 days after I've delivered Emily, and I'm looking very happy and relieved at the same time. But I'm not sure if you can notice, I do have a bit of a yellow tint in my face. Even though this is one of the happiest days of my life, the first one being with my firstborn, events coming up to it were a little bit stressful, to say the least. So I actually was diagnosed with gestational diabetes in pregnancy. So this was my second time that I had gestational diabetes, because I also had it in my first pregnancy. It was a bit of a shock in the first pregnancy because I didn't expect it. I did expect it in the second pregnancy because there is increased risk of developing it sort of when you have had it in your first pregnancy. So, but in addition to this, I also developed towards the end of my pregnancy obstetric cholestasis, which is the liver dysfunction, often linked to the hormones secreted from the placenta, and we'll talk about placenta in a great detail later on as well. That obviously meant that I had to have induced labor. My children were not premature, so they were full term, and that was lucky. And the good news was that they didn't have preeclampsia. So you will learn about preeclampsia today in my talk. Whilst I had quite a few other complications, preeclampsia was not one of them. So we'll go back to the very start, and we'll give you an outline of actually how pregnancy starts with embryogenesis. So I'd like to thank my talented honor student Claire Richards for providing these pictures to me. So this is part of the female reproductive system, and this is the ovary, where ovum or egg gets released, and it travels down the fallopian tube to meet sperm cells, or spermatozoa, which actually then implants, fertilizes the egg, and that travels down the follicular tube and gets implanted into the uterus. But during this travel from sort of meeting the egg to actually implanting itself into the uterus, there are quite a lot of changes happening. So following fertilization, zygote is formed, which is a single cell which contains fusion of sperm cells and egg cells. And this zygote undergoes a number of divisions until it forms morula. Morula is a structure that contains about 16 cells at this point that have been divided and containing both genetic material from the mother and the father. And then further on day 6, a blastocyst is actually developed, which contains all these cells, but as you can see, it's a lot more organized structure, and it contains outer layer of differentiated trophoblast cells. Now, remember this name, trophoblast cells, because we will talk about them a lot. they actually end up remodeling maternal vasculature to allow supply of oxygen and nutrients to the baby, but also they form this organ called placenta. There is this inner cellular mass within the blastocyst where the fetus will be formed from. And the rest of the blastocyst is actually filled with fluid that will form amniotic fluid that will keep the baby floating inside mother's stomach and uterus. So what happens then? So once we have the blastocyst form, it travels down fallopian tubes to reach the uterus. And these trophoblast cells on the outside of the blastocyst are quite sticky, and they secrete chemicals that actually attach to this endometrium, which is the lining, the mucous lining of the uterus. So once the blastocyst actually attaches to the endometrium, it travels down, and these trophoblast cells then start dividing and forming this layer of cells that travels further down to actually reach spiral uterine arteries of the mother. And trophoblasts, what happens then is that trophoblasts actually invade these spiral uterine arteries, remodel them, which is our next part of the talk. So this is the cross-section and the magnified section of the spiral uterine artery within the uterus, or the myometrium of the uterus. So in non-pregnant state, these spiral uterine arteries are just sitting normal. They're supplying uterus with the blood and oxygen and nutrients, etc. But in pregnancy, so once the blastocyst travels down to the endometrium and then further down to the myometrium, which is this part of the uterus, it actually starts to remodel these spiral uterine arteries by allowing trophoblast cells, which form the outer layer of the blastocyst that we talked about in the previous slide. And what this does actually, it allows for these spiral uterine arteries to remodel and to become elastic and to allow unrestricted supply of oxygen and nutrients to the placenta and then further to the baby. But what this means for the woman's body is that there are a lot of changes, so both cardiovascular and metabolic changes. Some examples of these include increase in maternal blood flow 100 times, which is substantial, as you can appreciate. Blood volume also increases 30 to 50%, and heartbeat increases 10 to 15 beats per minute to meet the demands of all these changes and the growing fetus. So then placenta develops from these trophoblast cells, and what placenta actually does is delivers oxygen and nutrients to the baby so the baby can grow in utero, and also removes waste products. But also another important function of it is partial protection of fetus from teratogens and pathogens in the maternal circulation, which is. I'd like to thank my other honor student, Ingrid Anneman, for providing this picture to me for this presentation. What is interesting is that placenta is actually an endocrine organ. What this means is that it actually secretes hormones to substantiate the development of the fetus. So the hormones that are secreted include estrogen, cortisol, and human placenta lactogen. So what these hormones do, while they do wonderful things and enable pregnancy, really the progression of pregnancy, they also have the function of inhibiting the insulin and the work that insulin does within the body. So insulin is not very effective anymore in utilizing and breaking down glucose and metabolism. So that means that women actually have to produce more insulin from their pancreas, which is this little leafy structure underneath the liver. But in some women, this is actually not managed very well. The body can manage this excess insulin demand, and this develops into gestational diabetes mellitus. Gestational diabetes occurs only in pregnancy, and it is a major concern nowadays, particularly because there is increased risk of obesity nowadays with the changes in the lifestyle, and the number of women actually having type 2 diabetes as well before pregnancy is increasing too. So currently over 21 million women have some form of diabetes in pregnancy. So we can easily say that hyperglycemia, or the presence of high blood glucose in pregnancy, is actually one of the most common complications in pregnancy. And this complication in pregnancy does lead to increased size increased baby size, because actually the glucose passes through the placenta very easily, and it can be easily absorbed by the baby. However, this leads to sort of increasing the size of the baby, but it's not a good thing actually, because there is fat around the organs can be formed, and other complications as well. And in fact, what we've been seeing over the last 10 years is a 2 to 6-fold increase in the incidence of gestational diabetes. So what are the complications of gestational diabetes? A microsomia, which means big size of the baby, that can cause complications during delivery, such as shoulder dystocia, but also other complications such as preeclampsia. And preeclampsia is the focus of this talk and my research as well. In addition to preeclampsia, I also research diabetes as a risk factor for preeclampsia. So what is preeclampsia? Preeclampsia is the leading cause of maternal and fetal death in the world, and it affects about 5 to 8% of pregnancies. The number of women experiencing preeclampsia in pregnancy actually is on the rise, because there is increased number of women with obesity and diabetes. Also, after preeclampsia, or after the pregnancy has been finished, women are at increased risk of developing future cardiovascular diseases and also diabetes, and 2 out of 3 women will actually die from cardiovascular disease. Cardiovascular disease in general is the biggest killer in the world, accounting for 31% of deaths, even more than cancer, but these women are at particular increased risk in the future, so they need to be monitored closely. And knowing all of this, it is shocking that we actually don't have any effective treatments or effective monitoring strategies for women who have preeclampsia in pregnancy and afterwards as well. So this is the area that we focus on. So preeclampsia still kills over 70,000 women and 500,000 babies every year. Most of these deaths actually occur in developing countries, and this is because women don't have access to care so easily, and they live in remote areas, the healthcare systems are not very well developed, etc., and that leads to fatal consequences. Short-term complications from preeclampsia during pregnancy include preterm birth, and this is before 37 weeks of gestation. HELP syndrome, which is the hemolysis, elevated liver enzyme, and low platelets, very dangerous syndrome that can lead to organ damage and other complications as well. Eclampsia. Eclampsia are seizures, which can also be fatal. Stroke. Neonatal respiratory distress syndrome is another short-term complication associated with preeclampsia. And long-term, like I said in the previous slide, it's diabetes. Women have increased risk of diabetes, cardiovascular disease, and stroke as well. It is possible that some of these women already had sort of underlying risk factors for developing these diseases later on in life that actually get unmasked during pregnancy. So actually, whilst pregnancy is a stress test for the body, it's also an opportunity for intervention early on to prevent any complications and death further in the future. To make things even more complicated, there are actually at least two, in fact, phenotypes of preeclampsia that we know of. We are still researching and trying to understand this condition better, but what we know so far is that there is early onset preeclampsia and there is late onset preeclampsia. And these two types of preeclampsia are subdivided according to the time of gestation of their diagnosis. So for example, early onset preeclampsia is diagnosed before 34 weeks of gestation, and it is normally associated with difficult complications in placental development and also intrauterine growth restriction so small babies, in fact. Whereas late onset preeclampsia, which actually consists 80 to 90% of preeclampsia cases, a lot more cases, women would have this type of preeclampsia than early onset preeclampsia. Whilst it's potentially milder, it actually occurs after 34 weeks of gestation. And often it is not associated with placental complications and restrictions in the growth or the development, in fact, but still there are some overlapping features between the two different types of preeclampsia. Hence, it is not so straightforward to identify one or the other type. Late onset preeclampsia, what we know so far, is often associated with sort of underlying complications such as diabetes and obesity and other vascular complications in pregnancy. And I'd like to thank Ingrid again for providing this slide for me. So how does preeclampsia develop? What we know in terms of sort of placental development, when placenta is actually involved in its development, is that if you remember from our blastocyst slide, where we had our trophoblasts that travel down the sphirouterine artery to remodel it. So in normal pregnancy, this is done properly, and the sphirouterine arteries are remodeled. They're wide, they're elastic, and there is unrestricted supply of oxygen and nutrients to the placenta and the baby. However, in preeclampsia, this process is actually restricted, leading to a restricted supply of oxygen and nutrients to the baby, potentially a restriction in the growth of the baby, but also other complications, because what these trophoblast cells have to do, and these are fetal cells, if you remember from our blastocyst that we talked about earlier on, they have to remodel this artery, and by remodeling, they actually have to interact with endothelial cells. Now, endothelial cells are cells that line the blood vessels and our vasculature through which blood actually flows and supplies various organs, including placenta and the baby in pregnancy. So these endothelial cells, if they're not functioning well, because there is an underlying vascular risk factor that women have without ever knowing it, that can cause this inappropriate development of the placenta, or also inappropriate sphirouterine artery remodeling, as we call it. But if the fetal cells, on the other hand, as well, are not functioning well, this can cause problems too. And therefore, a lot of our research will actually focus on identifying factors that can influence the function of the trophoblast cells, but also the function of the endothelial cells as well. So before we go on, I'd like to pass you on to Laura for any questions that you might have, to break down the talk into two sections.
For a fascinating introduction into pregnancy, Lana. We've had a lot of questions come through in the Q&A and also from the event registration, and I'll try and get through as many of these as time will permit. Our first question is from Deb, and she wants to know, do we know what causes preeclampsia?
That's a great question. Thank you very much, Deb, for asking that. So a lot of the mechanism and the sort of the causes of preeclampsia are unknown. We are still trying to investigate and understand why this happens. So that is the big problem, why we haven't really been able to come up with very effective preventative treatments or curative treatments, and also monitoring strategies. But what we do know is that diabetes, for example, is a risk factor, and that can lead to, that increases the risk of preeclampsia. So for example, women who have pre-existing diabetes before they become pregnant, they can have up to 4-fold increased risk of preeclampsia. If woman has a higher BMI as well, that is a risk factor too. Sometimes age can be or sort of older age, but it's not significantly increased risk, but it is associated with the risk of preeclampsia. And other autoimmune diseases, chronic hypertension, for example. It is interesting that first-time mothers actually have sort of background increased risk of preeclampsia rather than the second-time mothers, for example. So there is something we know about it, but there is also a lot that we don't know that we need to find out going forward.
We have another question that you touched upon there. The question is there a higher risk for people who are older? And if so, why?
So There is an increased risk potentially of preeclampsia as you get older, and age is one of the risk factors too, generally over 40 years of age. We don't really know exactly why this happens. It could be to do with the vasculature and sort of more stress that body has gone through an aging process itself. But again, it's not very well understood. And we are trying to sort of understand how these aging processes can actually affect and lead to it. Again, coming back to vasculature, if there is a sort of underlying issue with the vasculature, endothelial cells are not working so well, etc., that could be one of the reasons too.
We had 2 questions from Marina. She wanted to know, what does a 100-fold increase in blood flow mean? Do you mean that it circulates faster? And also, could you clarify what you meant by 2 out of 3 women who have had preeclampsia will die of cardiovascular disease?
Sure, no problem. Thank you, Marina. So 100 blood fold, actually more blood is produced to substantiate the development of the fetus. So the body has to go through all these changes, more hemoglobin in the red blood cells, etc. So it's a fascinating machinery, really, and what women's body is going through. In terms of future, so as I said, cardiovascular diseases are actually the leading cause of death for everybody. So the 30% of deaths come from cardiovascular complications. So these women, for women, cardiovascular disease anyway is the #1 killer too. But because preeclampsia can actually cause this inflammation and oxidative stress, and we'll go in the second part of my talk about these sort of stress factors that can remain after pregnancy and therefore lead to damage to the blood vessels, etc., that can lead to cardiovascular disease.
We've got a question from Kathy, and she wants to know, what are the first symptoms of preeclampsia to watch out for?
That's a great question. Very difficult. Preeclampsia is sometimes also very difficult to diagnose. It's not that straightforward. In some women, we have typical symptoms of high blood pressure. So high blood pressure is one of the compulsory, you would call them, symptoms. But in addition to high blood pressure, there has to be other symptoms as well. Otherwise, if it's only high blood pressure in pregnancy, that's called gestational hypertension. So in addition to high blood pressure, we also have organ damage, for example, kidneys or liver. So if there is any liver function test are showing sort of abnormal results, or for example, if there is protein in the urine where it shouldn't be, that means kidneys are not working so well, etc. Other symptoms in addition to high blood pressure could be headaches, swelling in the feet, around the ankles, and other organ damage. Cerebrovascular, for example. Platelets could be also affected in addition. Generally why we are working so hard to actually try and figure out the way to monitor women from the early pregnancy and to pick up preeclampsia early is because it comes on so suddenly. In a lot of the cases, pregnancy is going really well, nothing's wrong, and all of a sudden all this happens and women end up in the hospital, etc. So we are trying to actually find a way that we can monitor it from early on in pregnancy so that we can diagnose any changes or follow any changes early on without complications and sudden onset of severe symptoms and early birth, of course.
Thanks, Lana. I'll let you tell us about your research now, and we'll have some time for more questions at the end.
Thank you very much. Great. So we'll go on about the details of my research and the research of my team. Of course, it's a team of collaborators and students and postdocs that help with this research. So our focus is actually on trying to understand the mechanisms underlying preeclampsia. What happens early on in pregnancy with the placenta or with other cells that then leads to the development and manifestation of preeclampsia later on in pregnancy. Bearing in mind that the preeclampsia actually gets manifested sort of after 30 weeks of the gestation. Anytime after 20 weeks of gestation actually can occur, but in most cases it will be after 30 weeks of gestation. So we are actually investigating some novel proteins and microRNAs. MicroRNAs are small nucleic acids that regulate translation of genes into proteins. And proteins are very important because they are actually key for the phenotypical changes in our bodies. So whatever is the result of molecular changes actually comes through changes in the protein expression or differences in the function of those. So we are using placenta as a model to understand any molecular changes and how these can be leveraged and used for developing, for example, tests. Inflammation being one of the very common underlying processes in preeclampsia as well. So we are understanding how these proteins and microRNA can affect inflammation, lead to development of inflammation, etc. Angiogenesis is the vascular development. So obviously there is a lot of angiogenesis happening in the body during pregnancies because extra blood is needed to supply fetus. Endothelial dysfunction, so cells that line the blood vessels that we talked about as well. We want to understand how these proteins and microRNA affect their function. And also cellular processes, because all these cells, trophoblast, endothelial cells, and other cells that are actually present within the placenta, and these include sort of other cells such as mesenchymal stem cells, immune cells, etc. They all have to undergo proliferation and differentiation, so maturation as well. And apoptosis is a cell death, really. So obviously there is increased cell death potentially in preeclampsia that we're trying to understand why this happens and what regulates these processes. So I just wanted to show you some of our work. So this is the cross-section of the placenta. So we took a little chunk from the placenta from a mother who delivered a baby, and placenta is a massive, big organ. So therefore, there's plenty of tissue to be investigated and used for research purposes. And what we did actually, we made little slides of this placental tissue, and we looked at the expression of trophoblast cells and also blood vessels or endothelial cells that line up blood vessels. So this is how placenta looks inside. It's actually composed of many of these chorionic villi, which are surrounded by trophoblast cells. And within these chorionic villi, we can see blood vessels as well, which are only shown on this picture. So for example, when we want to look at the differences in the expression between one or many proteins that we are interested in, we could actually look at the differences in the expression of these proteins in healthy placenta versus placenta that was from women with preeclampsia. And this picture just depicts an example of how this particular protein is actually overexpressed and increased in preeclampsia compared to healthy pregnancies. So because it's not so easy to get early placental tissue during pregnancy, the procedures are invasive and it's not something that we can do routinely. We're trying to actually develop models of the placental development in the lab using trophoblast and endothelial cells. So this is the work of my very talented honor student, Claire Richards. She actually was trying to develop these mini placentas in the tissue. So it's this is a 3D placental structures and composed of trophoblast cells, the first trimester trophoblast cells, that actually organize themselves into these spheroids or organoids. And you can see that we've stained them with different proteins that to show the nuclei of the cells or to show other features of the cells that are important in the development of these sort of mini placentas. And when we actually take those out of the culture and cross-section them, so we cut them in half or across, and we looked inside the cells to see whether we have different types of cells, and it's not just one type of cells, because it's this multi-hierarchical organized structure of placenta. We've actually realized that we have many different cell sizes within each spheroid or organoid. Some were bigger, some were smaller. Therefore, we had that heterogeneity in terms of cellular structure. But further work that I've done with Professor Varkiani from the biomedical engineering is to try and develop these mini placenta in a more dynamic way. So having sort of been able to kind of see the development and measure changes in real time as cells interact. So what we're doing at the moment, we're trying to co-culture these trophoblast spheroids that I showed you on the previous slide, together with endothelial cells, so we can mimic what's actually happening with the placental development and spirouterine artery remodeling, where these cells actually interact together. So this is a fascinating platform that Professor Rakiani actually has in his lab, where we can put many different cell types on this chip. So it's almost, we're building placenta on a chip. And we can put these endothelial cells in the middle, for example, and trophoblast cells on the side, and then add other vesicles or other cells that are of interest to us, as well as other treatments, or collect supernatants, which will contain changes in the protein expression that we can use as biomarkers. So all of these then represent sort of more dynamic structure, and we are using this to elucidate mechanisms affecting early preeclampsia and also early placental development, but also to test some treatments that we could potentially develop and use either to prevent preeclampsia or to treat preeclampsia. A specific protein that we've been interested in elucidating its role in preeclampsia is called FKBPL. So we started this work 5 or 6 years ago, and we realized that all these processes that have been well established in preeclampsia, including impaired vascular response or anti-angiogenic response, which means restricted vasculature, also abnormal trophoblast invasion or abnormal trophoblast function, oxygen changes when there is sort of restricted vascular development, then there is obviously low oxygen tension, and we call this hypoxia or reduced oxygen concentration, and inflammation or autoimmune system play a big role. And actually this protein that we are interested in plays a role in all of these processes. We have been exploring its role as a biomarker, but also potentially as a treatment target for treatment of preeclampsia too. So we filed 2 patents in relation to this work, which are currently being commercialized and validated and developed further as new tests of preeclampsia. So what we propose with our biomarkers, and we have developed an advanced algorithm, not just including one protein, but a couple of proteins and other clinical characteristics. We can identify changes in these, in the level of these proteins in combination with clinical features such as blood pressure or sort of proteinuria or etc. And then when we measure this sort of towards between 2nd and 3rd trimester, which is still sort of before preeclampsia develops, we see the increased levels of these proteins. And then when preeclampsia is actually diagnosed, the ratio goes down. So if we can detect these changes early on before full-blown preeclampsia occurs and manifests, we could potentially do something about it and reduce premature births and deaths, even if it just means monitoring women closely and giving symptomatic relief therapy as well can prevent complications. So because most of these of the deaths associated with preeclampsia occur in developing countries due to the poor access to healthcare and monitoring, etc., the best way to actually translate these tests into something that can be beneficial for women and babies all around the world is to develop a digital health solution, which means that remote monitoring, something that women can actually do from home and be monitored remotely by the clinicians or the healthcare team who can then intervene if they need to and if they see any changes in various parameters that could stipulate that there is something going on that could indicate preeclampsia. So our proposal is to sort of be able to connect many monitors and also point-of-care devices. Point-of-care devices means it could be like a pregnancy test in the form of a pregnancy test, but it could be lined with various sort of proteins that we think could have a role in preeclampsia. So therefore at home, patients can do their own testing and upload this information onto the app or the cloud, where clinicians can remotely check this and then alert patient if there are any changes that could be indicating the development of preeclampsia. And then we've been doing this work at UTS with another collaborator of mine, Professor Jin, and he has a fascinating platform as well, point of care test platform that can be translated for many different diseases and testing and monitoring of many different diseases. We've been working on actually translating our biomarkers into this platform that then can be incorporated under this digital health umbrella. This work is still early stages, but it is promising. So then shifting gears, we want to monitor women in pregnancy and also detect if there are any changes, which can prevent serious complications. But actually, what are the treatments? Can we actually treat preeclampsia? How can all of this be prevented or treated? And like I said, there are no effective treatments so far, but what we do have is a limited therapies. So for example, aspirin, low dose aspirin and calcium are used in some cases in high risk pregnancies, for example, that we know have underlying high risk of developing preeclampsia, such as, for example, women with diabetes. However, aspirin has been shown to reduce the incidence of preeclampsia, but mainly of early onset preeclampsia. So that is a lot stronger for that and not so strong for late onset preeclampsia, which, if you remember, actually consists 80 to 90% of preeclampsia cases. Calcium is also used as a supplement for women who have particularly low calcium level in the blood. So if they have normal calcium level, it might not work so well as if they have lower calcium levels. So these are two options that we currently have, and a lot more is in development. And then what happens when preeclampsia gets diagnosed? There is high blood pressure, there is organ damage. Then all we can do really is symptomatic relief. So we can give women antihypertensives to bring the blood pressure down. Magnesium can work well for that as well, just to kind of get pregnancy to term so that we can prevent this preterm birth, which is associated with complications. And often we don't have a choice but to deliver the baby and therefore the placenta, which in most cases would stop preeclampsia. But again, there is that residual risk of future complications. So we've been working on this novel treatment strategies, which consists of using mesenchymal stem cells and their secreted vesicles, which have a lot of beneficial effects. This work is at preclinical stage, so it hasn't been tested in human, but it has been tested in various models of the disease. So mesenchymal stem cells are probably one of the widest used stem cell type therapies at the moment, and they're one of the safest stem cell therapies at the moment. And many clinical trials in different diseases have been carried out with these type of stem cells. They can be isolated from adipose tissue, placenta itself, or bone marrow. And what we've kind of been realizing over the last few years is that these mesenchymal stem cells actually don't necessarily differentiate and form different types of cells, but rather secrete these vesicles called extracellular vesicles that contain all the proteins and microRNA and sort of all the genetic cargo that has all these beneficial effects, and therefore can improve the angiogenesis or can improve that vascular restriction that can happen in preeclampsia. It can reduce inflammation, and also it can act as an antioxidant as well, potentially reducing the symptoms and curing this condition. In preclinical models of preeclampsia, they have been shown to have beneficial effects and to be able to reduce high blood pressure and organ damage. And we have actually recently reviewed this in the current hypertensive report. So if anybody's interested, you can look at our paper on this. So what happens after preeclampsia develops and the baby is delivered and preeclampsia disappears? There is that residual risk of cardiovascular disease that women are, that 2 out of 3 women will actually die from in the future. So cardiovascular disease is the single biggest killer in Australia, but also worldwide, accounting for over 30% of deaths. So women who have had preeclampsia, after preeclampsia is gone and the baby is delivered a few years later, even within the first 5 years postpartum, they have 3.7-fold increased risk of developing hypertension and 2.2-fold increased risk of developing heart attack, for example, or ischemic heart disease. and 1.8-fold increased risk of stroke. So therefore, what we should be doing really when women had suffered preeclampsia in pregnancy, we need to make sure that we're following them really well, monitoring them closely in the future, and trying to prevent or delay or manage these diseases better, so there is no surprise. We have been working on this paper, which is also published on this study with collaborators from Queen's University Belfast and also Mayo Clinic, where we wanted to look at the overlapping mechanisms between preeclampsia, hypertension, and HFpEF, which is heart failure with preserved ejection fraction. It's a form of heart disease. So because we know that these women have increased risk of cardiovascular diseases in the future, we don't actually know what the mechanisms are. Again, coming back to the molecular levels and understanding what happens so that we can actually leverage this knowledge and develop tests or treatments, etc. And we have identified about 30 different mechanisms that could be overlapping, and therefore they are worth pursuing further and trying to sort of understand these links better and develop tests, for example, or treatments for that matter. But other work that we've been developing in collaboration with another colleague from biomedical engineering at UTS, Dr. Gentile, is trying to develop these cardiac spheroids. So similar to what we're doing with our placentas, developing mini placentas, here we want to develop mini hearts in the dish. So Dr. Gentile has this amazing platform that is capable of bioprinting these cardiac spheroids, and they're shown here. So it's, again, they would represent heart tissue by using blood from patients that can be converted into stem cells. And stem cells are pluripotent, so they can give rise to many different types of cells. And by using patients' blood, for example, to develop and make these stem cells, we can actually then make these stem cells produce other cell types, which are main or key cell types within the heart. And these include cardiomyocytes, endothelial cells, and cardiac fibroblasts. So these three cell types are very important within the heart tissue, and cardiomyocytes in particular, they compose most of the heart cells and tissue. So we mix these three different cell types in a little matricial tube, for example, and then they organize themselves and form these sort of heart, cardiac spheroid or mini heart tissue that we can then use to understand mechanisms again, or we can test some treatments to see whether certain treatments will work in a certain group of patients or not. And this is sort of just by using blood from women post preeclampsia, for example, post complications in pregnancy, so that we can understand if they have any, if they will have any complications, future complications of cardiovascular disease. And then when Dr. Carmine stained these cardiac spheroids, then you can see that their cardiac myocytes are actually in the red, and they consist most of these cardiac spheroids, and then the green are the cardiac fibroblasts, and the blue are endothelial cells, and they've organized themselves very similarly to what we would see in real heart. So it's an excellent model for trying to investigate the mechanisms, biomarkers, but also treatment strategies for women who are at risk of developing cardiovascular disease post preeclampsia, for example. So in summary, what I've sort of tried to give you an overview of pregnancy and our research and what we've been actually working on in my lab with many collaborators. So we are working on a new test for preeclampsia based on a novel mechanism that we think we can leverage to potentially early diagnose preeclampsia or monitor women from early on in pregnancy. We have developed, or we are developing actually, heart and placental tissues that are representative of a human tissue. And we are also trying to understand the mechanisms between preeclampsia and future cardiovascular complications so that we can develop better monitoring and treatment strategies for these women. And working on new treatments as well to treat preeclampsia in pregnancy, which are related to mesenchymal stem cells and its secreted vesicles. So this work doesn't happen very easily and simply and by myself. So a lot of collaborators are involved and students in this work, and I'd like to thank them all for all their effort and excitement to actually work on these projects, and many different universities and organizations here in Australia, but also abroad, are also involved. So thank you for listening and for attending my webinar as part of the UTS Science in Focus events. The webinar will be recorded and available on this link. So I'll take now some questions from the audience if there are any further questions.
Thanks, Lana. That was a really interesting talk, and we do have many questions from the audience. Our first question came from two different people, very similar question from Dana and Para. Are there any groups or subpopulations of people who are more susceptible to preeclampsia, and is there anything that can be done to prevent it?
Yeah, so the women with diabetes, for example, particularly with type 1 and type 2 diabetes, which has developed before pregnancy, not as a result of pregnancy, would have increased risk of preeclampsia. Also, some women with autoimmune diseases, or if they have chronic hypertension before preeclampsia kicks in, they would be at increased risk of developing preeclampsia. So in these women that we know, when they come in for their 12-week scan or early on in pregnancy to obstetricians, we actually they would normally be started on aspirin. So aspirin is the only really preventative treatment there is to potentially prevent in high-risk women preeclampsia. But again, it's not so effective. That is the problem, and particularly is effective for early onset preeclampsia rather than late onset preeclampsia. Other options, so what we are trying to focus on, we're trying to just increase that monitoring in women using low cost and remote monitoring. So just by monitoring your blood pressure closely, and most women wouldn't, because they wouldn't know, or they wouldn't be at a risk of developing anything, they could potentially pick up these changes. So it doesn't mean that everybody should be doing that, but if anybody is at increased risk, if they're tested and they have increased risk, for example, of developing preeclampsia, they should be monitored closely, for example, and that would mean that they could early diagnose it and prevent complications in preterm birth. Also, another way of trying to not prevent it but reduce the risk will be by not making sure that women do not put a lot of weight in pregnancy as well. So the weight is managed well. Traditionally, it's been thought that we can eat whatever you want in pregnancy, but as you can see from my talk, there are a lot of hormonal and metabolic changes and cardiovascular changes, so weight has to be sort of managed well, whilst obviously there has to be a certain amount of increased weight. If it's too much, that's also not good.
And are there any ethnic groups that are more likely to develop diabetes or preeclampsia?
Yeah, so generally, ethnic groups such as, for example, Aboriginal and Torres Strait Islanders in Australia, or indigenous groups, they tend to have increased risk, not just necessarily of preeclampsia, but diabetes in pregnancy and beforehand, and that could be sometimes linked as well to the increased risk of increased level of BMI, or also the sort of some genetic predispositions too. And if there is a familial link as well with preeclampsia, so for example, if a mother or anybody else in the family had preeclampsia in their pregnancy, then that could potentially be also a risk factor for preeclampsia in your own pregnancy.
We've got another question from two people that's very similar. Have you tested FKBPL in urine from pregnant women? Could this be done like a pregnancy test, or do you need to use blood for your tests?
So far, we've only done it based on blood. So we still have to obtain blood. We haven't really measured it in the urine. We don't know if we can pick it up in the urine. Urine, of course, will be the least invasive way of doing it, but because women are getting blood tests done for various things throughout pregnancies, especially at 20-week scan and 20-week checkup, then it wouldn't be necessarily an extra blood sample, but it will be sort of part of the regular monitoring.
Do you think that the increase in telehealth due to COVID-19 will help encourage the use of apps like the ones you've described?
That's a great point. It has actually come, it's coming to light with COVID-19. They're really promoting the use of digital health and telemedicine, and in fact, they have acknowledged that as a service that clinicians can actually provide and get reimbursed for, which is a big step forward for digital health, in fact. And I think now is the time when we realize how important this is. I mean, just going by my personal experience, when I had gestational diabetes in my both pregnancies, I had to go in every week or every 2 weeks, and that would have required half a day off work and two hours travel each way, and then 2 hours wait in overcrowded waiting rooms and areas just to see somebody for 10 minutes. So I would have much rather have done this from home, whereas I can still be monitored by a clinician and escalated if that needed to be, but actually sitting in the comfort of my own home or even from work.
Do you think there could be any risk of over-monitoring by nervous first-time mothers if they can do it themselves in their house?
That is very true, and that is one of the points that we have to acknowledge as I guess as a limitation, because everybody is quite nervous in pregnancy, especially first pregnancy, and everything is going well, etc. So yes, there is that, but again, clinical staff will advise what is the most optimal number of test or monitoring points that you have to report. For example, for blood pressure, could be twice a week. For blood glucose, for women who have gestational diabetes or other type of diabetes, they would have to monitor their blood sugar 6 or 7 times a day. That's a lot of pricks. So it's just how, you know, you have to do it, whatever the clinicians actually advise you to do it.
We've got a question from Alison, and she wants to know, how are microRNAs being explored in preeclampsia? Are you focusing more on their role as biomarkers or their biological roles in the development of preeclampsia?
So whilst biomarkers are actually generally used as a test to predict the likelihood of a disease or to actually diagnose a disease, they also give good insight into the mechanism and why this is happening. So most of the time they have biological role. In some cases it could be just an association and not necessarily a mechanism of the disease, but a lot of the time there would be a biological link as well.
There's a follow-on question from Glenna, and she wants to know, would you be transfecting cells with extracellular vesicles containing specific microRNAs to promote or demote specific protein translation?
So, yes, so what we do, generally we collect these extracellular vesicles from the secreted medium of mesenchymal stem cells, and then we add them onto the cells, but we can certainly manipulate the protein or RNA or microRNA expression if we find that certain microRNAs will make these vesicles more effective. That is also possible to do before we actually put them on the cells and test them.
Another question from Alison is, what is the reason for proteinuria in preeclampsia? Is it due to damage of the kidneys or the blood vessels in the kidneys?
Sure. So proteins shouldn't be present in the urine. And so when the kidney actually filters all of these proteins and puts them back into the body, essentially. So if the proteins are present in the urine, obviously a certain amount is okay, but above allowed amount, then that can that can suggest that there is damage to the kidneys. It could be many different causes of that, but it's generally either the glomerular damage, which is small cells, or the small cells within the kidneys. It could be due to many different reasons, but it could suggest kidney damage, obviously, that kidneys are not actually filtering these things efficiently.
Any long-term risks associated with preeclampsia for the baby, and has a genetic link been observed?
Yes, so actually everything that I talked about, including risk of future cardiovascular diseases for the mothers, the same risks are actually applicable to the offspring and the children as well. So there is increased risk of developing cardiovascular disease and diabetes, obesity, etc. for the offspring as well. Because of this, we call it fetal reprogramming, intrauterine fetal reprogramming that happens during pregnancy.
So we have a question here, and it is whether the stem cells that you talked about need to come from the mother or whether they come from an anonymous donor. And then also, are stem cells safe to give to pregnant women?
So stem cells, actually there is some evidence that mesenchymal stem cells in particular that we are interested in women with preeclampsia, they have actually impaired function. So this is the work that has been done by our collaborators at the Mayo Clinic, in fact, Professor Garovich and her team. And they actually take out and extract these mesenchymal stem cells from the adipose tissue following the birth of the baby. And they have shown by characterizing the function of these cells that these cells appear to have a different or impaired function. So hence, that might suggest that it would be better to use a healthy donor mesenchymal stem cells, or from a bank, for example, and more so using these secreted vesicles from the stem cells will be even better option in the future, because it will be a cell-free therapy. So any risks or problems that are associated with the cell-based therapies would then disappear as well. Again, we are a long way from knowing exactly how safe these will be in human trials, but we are. There are a number of preclinical trials that are showing promising results.
Excellent. We've got a question here from Marilla, and she wants to know, what is the difference between diabetes and gestational diabetes?
So gestational diabetes occurs in the second half of pregnancy. So women would develop it in pregnancy as a result of placenta secreting those hormones and body not being able to adjust, and generally disappears after the pregnancy is finished and the baby is delivered and placenta is out. Whereas other types of diabetes, such as type 1 diabetes and type 2 diabetes, they develop sort of outside of pregnancy in women who are not pregnant, and that's the main difference. Now, gestational diabetes can sort of turn into type 2 diabetes after the pregnancy in 30 to 40% of women who have had it, or sometimes in pregnancy, the type 2 diabetes can get unmasked because of the tests and the stresses that the body is going through that wasn't diagnosed before pregnancy. So that is the main difference.
We've got a personal question for you here. Given your knowledge about how scary the complications in pregnancy can be, were you worried at all about having babies?
Absolutely. I was terrified in both of my pregnancies, but I'm that kind of a person as well. I worry too much about things. But it was a worry, but I think it just kind of had to trust clinical staff and people who are monitoring me. I was lucky to go to the same hospital with my first and the second one, so they had my records and they knew of my history from the first pregnancy, so I was sort of monitored closely because I had gestational diabetes in my first pregnancy. So I would certainly recommend that if you are changing hospitals, to make sure that they have the full history from the first pregnancy, for example. So I was worried, and I just was hoping that the second one would be better, but unfortunately it wasn't. So now I'm done.
If you experience preeclampsia in your first pregnancy, what is the probability in your second and subsequent pregnancies?
Great question. There is increased risk. So if you had preeclampsia in the first pregnancy, there is increased risk for having it in the second. Again, you have to, your clinical staff and people looking after you, obstetrics and gynecology staff from the antenatal clinic and other health professionals, they should have that information, and then they would, if you have any concerns, by all means, speak to them, because they will manage you in the best way possible. So there is that link.
What are your thoughts on the placenta's medical benefits post-pregnancy? A very controversial field. A lot of it is unknown, so I couldn't really comment too much about it. Is there a genetic link to preeclampsia?
There is. So I mentioned that, I think, earlier, that if somebody in your family, your mother or aunt, would have had preeclampsia, there is increased risk. So if you're worried about anything like that, and if there is, I guess, preeclampsia around in the family, then you should most certainly speak to the doctor who's looking after you during pregnancy, and they would generally ask you these questions anyway as part of the first checkup. So speak to them, and they will answer all your questions and concerns you might have.
Are there any treatments to preeclampsia once you've been diagnosed with it?
That is the problem with preeclampsia, that we don't have sort of treatments that will cure the condition, and that it would cure sort of all these aberrant processes that are happening, and all these problems that are happening with the vasculature, with other cardiovascular effects. But the only option once somebody is diagnosed with preeclampsia is to manage the blood pressure with antihypertensives. And there are certain types of antihypertensives that can be used in pregnancy because they're dangerous for the baby. So there is an approved list of antihypertensive drugs which can be used for the management of preeclampsia, but they would only really manage symptoms until woman is ready to give birth, and the whole sort of strategy will be around trying to delay that preterm or premature birth, really. So this is an area that certainly needs to be improved and researched more, and we have to come up with better and more effective treatments to actually cure this condition.
You mentioned that diabetes contributes to an increased risk of preeclampsia. But does it work in the other way? If you have preeclampsia, are you at an increased risk of diabetes further down the line?
Absolutely. So preeclampsia increases the risk of future cardiovascular diseases, but also diabetes in both mother and offsprings. So there is a risk. So whilst diabetes is a risk for preeclampsia, and generally diabetes in adult life is a risk for future cardiovascular disease, it's all intertwined and interlinked, and a lot of it is unknown, but certainly the epidemiological link between preeclampsia and future cardiovascular and metabolic diseases such as diabetes has been established.
If apoptosis is reduced in preeclampsia, then will that impact on other disease states that are controlled by apoptosis?
So quite often, again, this is under research, and still a lot more that we don't know about it, but there is an increase in apoptosis potentially of trophoblast cells. So we are trying to actually prevent apoptosis from happening. Whereas, for example, in some other conditions such as cancer, we're trying to kill cancer cells. It's sort of the opposite of what we're trying to achieve here, prevent that apoptosis.
How does nature or nurture influence gestational diabetes and preeclampsia? What lifestyle choices can help with dietary deficiencies that may contribute?
Mm-hmm. Sure, very good question. The gestational diabetes is very much linked to obesity and high body mass index. So, and there are some studies which have shown that in women who are at risk, for example, of gestational diabetes in general, for example, if they have had gestational diabetes in the first pregnancy, and there will be naturally an increased risk of it in the second pregnancies if they lose weight or manage weight better before pregnancy, and bring in diabetic patients, for example, if they bring their glucose control within the required and desirable limits, that reduces the risk in preeclampsia. So by managing your blood glucose and your body weight well, and eating healthily and balanced diet with allowed exercise, preventing that sedentary lifestyle, all of these things have shown to improve healthy outcome of the pregnancy.
Do you know anything about the implications of COVID-19 on a fetus's growth and development?
It's a very hot topic at the moment, and unfortunately there's not much evidence coming through. So far, it has been reported that there is not necessarily an increased risk than any other pregnancy, but this is still to be confirmed and firmed up as more pregnant women actually have it, and we have better evidence to support these findings. I mean, in terms of influenza, for example, influenza can be, or the flu, for example, it can be dangerous for the baby and the pregnancy and the mother as well. So pregnant women are encouraged to have their flu vaccines during pregnancy to prevent themselves from the flu. But in terms of COVID-19, so far, the reports are saying that it's not any more dangerous than in the general population, but it's still to be determined.
You talked about mesenchymal stem cells, and they are different according to their different types of tissue, their stage, and their proteomic or genomic profiles. So what types of cells were you planning to use?
So we've been working on, in particular, on bone marrow-derived mesenchymal stem cells. Bone marrow-derived mesenchymal stem cells actually have been the most widely characterized, and they've been used in clinical trials for patients with different conditions, not necessarily in pregnancy, but sort of outside of pregnancy, for cardiovascular diseases, respiratory diseases, etc. And they're sort of the gold standard ones that we will refer from. But a lot more work now has been done with the umbilical cord derived mesenchymal stem cells. They're the second, I would say, most popular ones. And then also adipose tissue and placenta itself. So far we've been working with bone marrow, but we are planning to also use the placental derived stem cells in the future research, and adipose derived stem cells as well.
Well, thank you for a brilliant talk today, Lana, and thank you to everyone who attended. So thank you again for joining us, and have a great afternoon.
Thank you, everyone.